I hope your diagrams are all dazzling. At Diagram Dazzles, this magic will definitely happen. Yes, for today's session, we'll learn yet another diagram from the NCERT second year biology, that is mature dehisced anther, a transverse section. So here it is dehisced. Unlike the young anther, you can see the anther ruptures at maturity to release its pollens. And here is a detailing. The outermost is the wall layers, out of which the epidermis is the first. It is thin and single layered and it continues to divide. Next one is endothecium. This is unlike what you see in the young anther. It is completely differentiated and it has got bands of thickenings. So you'll have to draw it squarish and with the bands of thickening. Next is the middle layers. The middle layers importantly are thin, and that is why they are easily ruptured as the growth happens within the microsporangium. The tapetum also undergoes disintegration. Important parts are the stomium. Stomium is a modified part of the epidermis where the dehiscence actually happens. It is the thinnest part of the epidermis in fact. So here you can see the dehisced wall and across it is the stomium cells. And of course, last is the pollens, which you will have to represent within, and they are matured. So what are the tricks? The first one, draw an outline. The outline consists of 2 ellipses and 2 circles. Yes, and here develop the association. Association is butterfly with the broken wings because you are going to show the dehiscence. Then of course do the detailing. So let's start. The first one, remember, you will have to draw 2 ellipses but keep a distance of 3 cm apart. And then beneath that, draw 2 circles, not a perfect one required, but draw 2 circles which are 2.5 cm apart. Remember, the outline will appear like that of the butterfly of a typical anther. So here is the outline. Now develop on this outline. The first part you will have to keep it for the wall layers. For the first 2, upper 2, seal it like a dome-like structure, and then the lower ones also draw the wall layers, but here you will have to draw the groove. Remember, we drew the same for the young anther as well. Next comes the game changer. Here in the next part, you will have to trim this outline because we are drawing a dehisced anther. So the microsporangia at maturity undergoes dehiscence. For this, you will have to erase the part of the ellipses and the circles which you have drawn. So erase the part wherein they face each other in each of these anther lobes, as you can see here. So for both the ellipses and the circles, you will have to gently erase the outlines wherein they fuse, the microsporangia in fact fuse. To make it easy, I am highlighting it with red. So these are the areas which we have erased. And once it is completely done, you can see it like this. After that, we'll start with the drawing of the tissues. For that, I'll represent the diagrams on one part. The same way you can draw it for the others. So start with the outermost layer, that is nothing but the epidermis. Draw thin cells, small cells in line, and then you will have to do it for both of them, the upper part as well as the lower part. So once it is completely done, it will appear like this. The next region, as I said, is very important, and that is nothing but the stomium. So stomium, you don't see it in the young anther but appears in the mature anther. So draw a slightly mango shaped or spindle shaped structures with partitions like this. Yes, so do the same for the lower lip cells or the stomium, and this is where the anther dehisces. Remember, there we'll have to keep a gap in between. Next, the endothecium, draw large squarish cells all across, both towards the upper part of the epidermis as well as on the lower side. Importantly, the next part you will have to represent is inside the endothecium, the hygroscopic thickenings, the striations which you can see there. So draw it, and after that go ahead with the middle wall layers, which are thin and flat. Remember, the middle wall layers undergo rupture, and hence we are not going to draw it across in completion, but keep to the outline of the ellipse and the semicircle below, and then draw it like this, as if it is ruptured. So once you have done with the upper side, also do it for the lower side. Remember, it should be thin rectangular cells, preferably drawn very light. After you have done with, come to the centermost region where you will have to draw a connective. Draw small cells squarish and then darken it. After that, fill in the entire region at the center by means of polygonal cells. So they should look very different from the middle wall layers since they are polygonal in nature. Then the filling is done with the pollen grain. First draw the outline and then gently draw the X sign. As you can see here, spiky X signs are drawn across the pollen grain. So fill the complete chamber, that is the 2 microsporangia which have fused now with the pollen grain, and some of the pollen grains coming out. With this, your diagram is complete on the right side. Do the same thing for the left side, and when it is completed, voila, it will look like this. So this is the TS of the mature dehisced anther. Then put the labelings, the wall layers, chromium, dehisced wall pollens, and then the connector. Yes, thank you for watching.